Hi, Yoli.
Hey, nice to meet you.
Yeah, can you hear me?
I'm okay. I'm okay.
Great.
Yeah, that's
Everything,
great. That's great.
everything is okay.
Okay.
Your presentation is good.
Yeah, so I sent you a record presentation, but I was told that it doesn't work. So I will be happy to talk directly.
Great. We are waiting for this.
So that will be good. Okay. So what is your time now? It's in the afternoon?
Now it's 3 p.m. in Israel.
3 p.m. Okay.
Yes. We wanted to find the
Okay.
time that is also good in the US.
Okay.
Yes.
It may be
So,
early morning
after
from
this-
Jeffrey, but it's okay. No, Jeffrey.
Okay. So now it's Jeff's morning, right?
What?
Yes, it's 7 a.m. here.
7 a.m. Okay.
And he's in the office.
Yeah.
It's because it's quiet.
Okay.
It's the best time for work, no?
That's right.
Hey, Eric.
Hey guys.
Hey.
Eric, we are on time. Okay, we have everybody participating on time.
Great. Okay, perfect.
Thank you for the flexibility anyway.
No problem. Hello to everybody else. Good to see you.
Hey, Eric.
Sharon, 2 more minutes and I will start. What do you think?
Okay.
Yeah. Sharon?
Sure. We can start. We
Oh,
have over
Christina is here.
40 people already, so we can just start.
So since I'm starting with the introduction anyway, yes. I'm starting with an introduction on the participants in the seminar and some activities of the consortium, so people can join and they will not miss the first talk. Okay, so it's a big honor to have you holding this live webinar on black soldier flight genetics from science to industry. We are very happy to host here 3 global leaders and major players in the BSF consortium globally. We have here Professor Yongping Huang from Chinese Academy of Sciences, Professor Jeffery Tomberlin from Texas University, and Dr. Eric Schmitt from Protix. So after, just a minute. After my short introductions on the activities and operation in the consortium, Professor Yongping Huang will talk about molecular genetics of black soldier fly. Then Jeffery Tomberlin will talk about the black soldier fly, non-traditional model for basic and applied research. And Eric Schmitt will talk about selective breeding in black soldier fly, implications for the industry. Between the presentation of each one of them, we will obviously have time for questions. And I think you can also use the chat for asking questions. So since we saw that we have in the audience participants which are not part of the consortium, I wanted to start with a short introductions on what we do in the consortium and who we are. So the Israeli BSF consortium is supported by the Israel Innovation Authority, the IAA. It has been established to promote domestication and to accelerate the development of circular economy solutions based on thoroughly crafted BSF farming. We are doing that in joint and multidisciplinary research, combining different research fields, including entomology, genetics, ecology, microbiology. We are trying to generate large body of research across the value chain of the species. Our activities include different research field.
Yoni, if you're changing slides, okay.
Come again? You see my presentation?
Yes, we see now our activity. Slide 3.
Yes. Yes. So some of the activities of the consortium, including mapping of waste and industrial ice cream as BSF feedstock ingredients, screening optimal rearing conditions and doing that in different scales, establishment of BSF colony and monitoring the stability across generation and preventing spread of pathogens. We are also evaluating the performance of high quality BSF protein and FRAS in animal feed, agriculture, and energy. And all of these activities, together with others, is being collected and gathered into BSF data storage that is aimed for machine learning tools for effective BSF rearing and also in the future recommendations for rearing and reproduction and all of the different steps. So just to mention that we are not developing in the consortium, we are not developing products, we are developing fundamental technology in a collaborative manner. We have in total 13 participants in the consortium. 8 of them, including BugEra, are private companies, and we have 5 academic groups. And the activity that I mentioned earlier is divided into work groups subjected into the relevant field. The first working group is focused on waste to feed, trying to focus on nutrition, testing different waste, and developing the crucial and the correct requirements for rearing, and trying to optimize the development of the larvae and to stand in industrial maintenance criteria. The second working group is focused on colony maintenance and optimization. This is where BugEra and other companies are active, and I will elaborate on this in the next slide because this is also the focus of today's webinar. And the third working group is focused on post-harvest processing and different aspects of it, including criteria of and characterization of protein and chitin, different trials of animal rearing, and also research on FRAS as potential biofertilizers and source for energy. So as you can see, these different working groups are sharing data and knowledge, and all of it is being gathered to the AI-based data management that is supposed in the future to supply recommendations. And you are welcome to visit our website and our LinkedIn page for more information. As I said, we are BugEra and other companies and academic groups are mainly active in working group 2. Some of the activity that we are doing and is relevant for today's seminar are monitoring and generation of SNP map for collecting data on genetic diversity. We are mapping this gene using RNA-seq from different developmental stages and different tissues. We are generating a transposase-based system for genetic engineering. In addition, we are tracking simulation of colony collapse that can be achieved due to genetic diversity loss using tools of selective breeding, which will also be discussed today. Two last additional topics are RNAi, that is a non-genetic gene silencing method. We are trying to develop this technology. And lastly, we are tracking pathogen. We are trying to identify them and to characterize them to avoid damage to our colonies. So just briefly to present our speakers today. We have here today Professor Yongping Huang from the School of Environmental Science and Engineering at the Chinese Academy of Sciences. He is, together with his students, is studying the molecular breeding of black soldier fly, and is also involved in research that is relevant for bioconversion. His lab is developing a platform of genome editing and transformation using generation of numerous BSF mutants at high efficiency. He's also the leading authors of many novel papers on BSF genome editing that was published in the recent years. Next, we will have Professor Jeffery Tomberlin that will present his research. He is a Professor, Alcalay Research Fellow, many titles. Presidential Impact Fellow in the Department of Entomology at Texas University. He is also a PI of the Forensic Laboratory for Investigate Entomology Sciences, also at the Texas University, and a director of the National Science Foundation Center for Environmental Sustainability through insect farming, and is one of the pioneer researchers in the fields of black soldier fly in different aspects, including reproduction, rearing, bioconversions, in independent research, and also in numerous collaborations that he has. And he's studying species interaction on efferent resources, decomposing plant material and animal waste, focusing on black soldier fly and insect as food and feed. And the last speaker is Dr. Eric Schmitt from Protix. Eric has a bachelor degree in economics from Reed College and a PhD in statistics from Huang. He's studied different aspects of insect that are relevant for insect industry, and it has been published in different international peer-reviewed journals, and is currently working as the director of R&D at Protix, which is a market leader in the production of insect-based ingredients for feed, including fish, chickens, pigs, pets, and other sources based on larvae of the black soldier fly. So I think we are ready for the first speaker, Professor Yongping. I will stop sharing, and the stage is yours.
Okay. So I will start to. Can you find my slides? Hello?
You want me to share your presentation and you will present it?
My presentation here, my slides here. Can you find my slides here?
You need to press on the green button, the share screen.
Yes. So I share the. Also, you cannot see my screen. Share my.
do you see the green button that said share screen?
Yes, I'll try. So can you find that?
I can open your presentation, and you will tell me when to continue, okay? If that's what you prefer.
Okay, yes, that will be great. Okay.
Okay, just a minute.
Okay, you can help me to share.
Okay, can you see it now?
Yes,
Okay.
I can
Just
see now.
let me know when to skip, when to continue, okay?
Okay, I will tell you to help me to. So, can you find that?
Yes, do you see it? Now you see my screen?
Yes, I think, but it's not big enough. I'm trying to make it bigger. Okay, I can find it. Okay, so can I start?
Yes, go ahead. We lost you, Professor Yongping. We lost you. Cannot hear you now.
הנה, יש לך את המצגת המוקלטת שלו?
לא, זה לא, זה לא עבד.
זה לא עבד? או כנראה לא תקשורת. So maybe we'll move to the next speaker,
Yeah, that's
and then we'll see when you join us.
שלח לו הודעה עם הצ'אט, והוא יראה את זה.
Hello.
Yeah.
So it says, okay, please go back to the first page. So, okay, can you hear me now?
yes, you can start. Go ahead.
Okay,
Please.
I can start. Okay, so dear friends, I'm very sorry for the internet problem. I cannot start from immediately. So thank you very much for Yuli and Uni to organize this online seminar to let me give a talk. So I'm very happy to see Jeff and Christina also in the meeting site. So these friends we meet have not met for many years because of the COVID. So tonight I would like to talk about the molecular genetics of black soldier fly. I moved from Shanghai Jiao Tong University. This is a very good university in China. From this March, I don't work anymore in Chinese Academy of Science after my 60. I will be very happy to invite friends to visit me in the university. This work has been done by many of the collaborators. We have these friends from the previous my research institutes, and also the friends like Professor Huang, she's from Central China Normal University, and also Professor Jibin Zhang and Yu Zi, that's Yu Niu. So Ziyunio is old friends of the many blank soldier fly people. Professor Ziniu Yu, he brought me into the blank soldier fly society. So I will be very happy to tell him that we have this seminar tonight. So next one. So I think most of the friends are very familiar with these insects, the blank soldier fly. Recent years, the blank soldier fly is become a very hot famous star in China. And for each year, we will go to many meetings in different places. Friends talk about the blank soldier fly, how to use these insects to treat the organic waste. You know, Chinese like to eat, to cook, and when friends come, we always supply enough food, more than enough, to invite friends. So the blank soldier fly could be a very good candidate to consume the remaining of the food. We, in most of the cases, we use the 13 star, 14 star, and 15 star. So this in the time is about 8 to 10 days. We could finish one cycle of the treatment. So during the in the treatment of the in the application of blank soldier fly in China, so we analyze that we need to solve many problems. For example, the blank soldier fly breeding for higher yield and quality. Before that, we have very few species is domesticated for application. So the next one is the blank soldier fly treats the target is organic waste or livestock manure. Young generation don't want to do this work anymore. So we need automation system. And the third problem is that we need to develop the downstream products. At the moment, we only use blank soldier fly larvae and the frass of the blank soldier fly. It is too simple. The value is not really realized as it has. And the fourth problem, that is when we are trying to extend our blank soldier fly application to the government, they always ask us about the criteria. They ask us, do we have a standard criteria, or do we have the permission from the Ministry of the Agriculture? But we don't have. So facing this problem tonight, I just talk about the blank soldier fly breeding for high yield and quality. So next one, please. So tonight I will focus on the first, the platform construction, and the second, breeding of the blank soldier fly strains. In the first part, I will talk about the genome sequence, but it's already the paper published. I won't talk too much. The second is we are trying to explain the microinjection and also the genome editing and the transformation. So this paper is published recently in a special issue of Insights. Many friends contribute to this special issue. The second about the breeding BSF strains. So we focus on three major directions. The first is about the ecological properties. And the second is about high yield. And the third is about the high quality. So tonight, I will just introduce about these two items. Next one, please. So about the blank soldier fly genome. So at the moment, we have two published, two versions published. The first version, our group and friends like the Jeff, we published together in 2020, is using the second generation of the sequence. We assemble a genome about 1.1 gigabits, and the quality is quite good. But it's very good. That is 1 year ago, the friends of Chris from the Oxford University then published the second version of the genome assembled using Parker-Bell. The quality is much more improved. It helps us a lot for in the gene function research, because the sequence the quality is good. Next one. So compared with the other diphtheria insects, the blank soldier fly has a larger genome size. It's about 1.1 gigabits. The other diphtheria insects, their genome is in general is much more smaller. So we guess that is a blank soldier fly when they need to live in a very dirty environment. They need to get the ability to adapt to this kind of the environment. So we do find many gene duplication and the genome size is increased. Next one. And for example, here is we just show the expansion of the immune relative genes in blank soldier fly. So here is we list the microbial recognition, signaling, and melanization, and also AMP genes. This AMP genes is antimicrobial peptides. So the number we announced is much more than other diphtheria insects. So we also get some of these genes is cloned and expressed and tested in different insect model. Next one. Here is just show just one example about detoxification and chemoreception. So for the detoxification, just give one example about the P450. So we know that all of these genes are relative with detoxification. So the gene number of from the BSF is much the number is much bigger than other insects. Next one. So next part, I will explain about the microinjection of the embryo of blank soldier fly. We trained our students. We had a microinjection system for silkworm more than 20 years, but still, when we transfer this technique to blank soldier fly, it still takes us quite a few months to adapt to the microinjection. Because at first, the egg is very easy to be broken after the microinjection. The hatch rate is very low. After the training, at last, our hatch rate could reach more than 80%. So in this way, our microinjection system works. We can do the transformation and the genome editing. Next one. So here we just give an example. When we want to do the genome editing or the transformation, we need many parameters. For example, the promoter. Before we do this work, we don't have many available promoters in the blank soldier fly. So here is our students are very patient to find many kinds of different promoters. For example, here is the actin 5C. So the graphs shows that the EGFP is well expressed, driven by actin 5C promoters. Next one. And also is actin P3000. And also using Drosophila SP17. So these promoters also shows it works. It can be applied in the blank soldier fly. Next one. So here we identify the gonad-specified promoters. According to our experience in silkworm, if we use the actin promoter or the whole body, the organ available promoter, in most of the cases, the insect will die. So here we develop the gonad-specified promoter. So this promoter will allow us to express, to make the genetic operation just in gonad. So here is also so we identify this gonad-specific promoter. It shows it works well. And so next one. So we also use this p-base. So the p-base is we test about p-base. So this p-base, when we apply, it shows that its efficiency for engineering is become higher. And so then we don't use it. So next one. So here is so the another promoter used to drive Cas9. So this EXU drive Cas9 is also show the higher knockout efficiency. So at the right corner, right upper corner, we could find the two bands. One is 860, another one is 530 base pair. So these two bands show that the efficiency for the parting of gene knockout is at a very higher efficiency. Next one. So in addition to this actin promoter to drive this Cas9, we also use the promoter to drive sgRNA. We know U6. Also, we always use U6 promoters to drive sgRNA. So here is we use we identify U6 genes and to find the suitable parts for promoter to drive sgRNA. Next one. So here is to show that it's the U6 promoter is also very successful selected. So is the EGFP. We found that is the U6 we use the different ratio. It shows the quite efficiency to cars to knock out the genes. Next one. So next I just explain about the germline transformation. So we had the knockout system for it's planned it's quite high efficiency in our lab. Most of our students they can do this gene knockout. But if we want to keep the line stable, we need the germline transformation. Here is we use the system to select the X from the anterior to posterior parts. In most of the injection, if the efficiency is high, when we make the micro injection in posterior end, then we So we use the donor plus P base or XYP base. So in this way, here we can show that in the G0 generation, we could obtain the transformed EGFP in G0 generation. And the sequence also confirmed our transformation is successful insert into the genome. Next one. So here is we show that is the we want to know whether our Cas9 expressed in the blank soldier fly is effective or has no effect to BSF. So here is the result show that is the Cas9 is transferred in the blank soldier fly has no effect on BSF in the growth, in the pupillation and growth, and also in the larva reach. Next one. So here is also shows the G1 generation. So the G1 generation still we could detect the EGFP expressed in the blank soldier fly. So it means that it is inheritable. The line is inheritable, and also insertion confirmation is in the genome also is confirmed. And we also using this system we could get more strain. So we have many strains in our lab. We could show that is in the next generation still the EGFP is expressed. So, okay, next one, please. So the next part, I will explain about our breeding using this genome and the genome editing and the transformation system to make the breeding of the blank soldier fly. Here we construct the genome editing system. We select the white gene of the eye in the blank soldier fly. Next one. So the wild type in the right part, wild type of the blank soldier fly, the eye is black. When the his card wait, hello. Hello. The scarlet. The scarlet, when the scarlet gene is knocked out, the eyes become color. So this confirms that our system works well. because in the next generation of the macro injection, so we obtain a lot of the this kind of the mutants. Next one. So the next one, we are trying to construct a stable wingless mutants. We had this paper published in 2020 with genome sequence. It shows that the knockout of the VG gene is possible to make the adult lose their wing, but it's not a stable inheritable lines. Here we construct the the genetic transformed lines. So here we show that the wild type, they have wing. When the EXU drive Cas9, they have wing, and the U6 drive SGRA, they has wing. They have wing, but when the hybridization is made, the EXU Cas9 female or male is coupled with SGRA mutants, male or female. So all of the next generation then lost wings. Next one. So the wingless BSF so keeps normal development. So they just lost wing. Their body weight and their lifespan doesn't have any change. So next one. So it's they just show in the D part, they just lost the ability for the wingless BSF. They lost ability to copulate because we know that the blank soldier fly they need to fly in the space and male catch female. Then cause success to copulation to copulate. Next one. This is from now I will introduce about our work trying to build the construct this string with larger body size. So we select the PTTH, the hormone and the insulin pathway genes. We know the hormone regulates development of the blank soldier fly, and also the hormones also has such affection. Next one. So the PTTH, when we found the PTTH gene is expressed mainly in brain. So this is just normal, confirmed the research in the Drosophila. When we knock out of the PTTH, the larvae is become very long-lived. In the most of the cases, the wild type, then in about 40 days, they will have the pupation. But the PTTH mutants, even the 300 days, they don't make the pupation, and still we keep the many of the such kind of the larvae, so they can live much longer. Our students keep this long-lived string. They can mutate eclosion many times, but they don't the mutation. Next one. So because this one it just shows the larger body size, but it cannot be applied for food waste treatment. So we select the total. So the total gene so also has when the total gene is mutant, so the body size also show bigger, but it's could be last about 55 days. So this is also too long. It cannot be used for bioconversion. The next one. So we analyze the PTTH and the total gene when they are knockout. So we would like to know what the downstream genes is affected. So we do the gene expression analysis using the transcriptome analysis. So we do find some gene upregulated and some gene downregulated. So students are selecting these genes, the variation, and trying to analyze the function of these genes. Next one. And then we trying to get from the other way, because this PTTH and TOSO. The TOSO is downstream gene of the PTTH, but it doesn't work. So we're trying to select other genes like MPL2. So this MPL2 gene is negatively regulates insulin signal pathway. So we got this gene is from Drosophila. In the Drosophila, when the MPL2 gene is mutant, the adult's body size is become bigger. Next one. So here is we show that is the mutation of the MPL2 gene. It shows the body size is become bigger. So it is good that is this gene in the 4th star. The Larry is the mutant Larry is much bigger than the wild type, but it's in this 6th star. The body size is become the similar. So in this way, we could harvest it. We could harvest this Larry in the 4th star when the body size is become bigger. So in this way, it could increase the yield. Next one. So in addition to the breeding, we have I just show you a few results about the antimicrobial peptides application. So we have found many microbial antimicrobial peptides genes. So we express this gene and trying to test about the effect to the silkworm. Next one. When this gene expressed in the E. coli, we use this one and test it in the silkworm. Next one. So it has the effect to inhibit the bacteria. So this is 2 bacteria we selected. Next one. Another 2 bacteria we select for test. So next one. And also we express these EMP genes in the silkworm. We test this in vivo. So in the in vivo test, we successfully obtained transform line. Next one. And also we test in this bacteria the similar as the in vitro test. So it shows the antibacterial effects. Next one. Also these 2 bacteria. So next one. So I think that is my time will be finished. I just give a very few examples for our application. We have constructed factory in China to use this system. Okay, next one.
Please silence the microphone, please.
Next one. So this we design a very short system, for the application.
Zachariah, can you mute your microphone? Yanni, can you mute?
Sharon is an organizer. You can mute him, I think.
So this procedure is already applied in several factories in China. So I think that this system always works well. So in this one, so I just show that we use blank solidify. We could convert the organic waste that becomes resources, like the blank solidify and fertilizer. Next one. So at last, I would like to thanks to Professor Yu. So he brought me into this blank soldier fly society. He asked me to do the research. So I would like to appreciate Professor Xu Zhihong. He's a very famous scientist in China. He always support me and give me encouragement. And also Professor Guoping Zhao. He's also an academician in China. So he's a very good microbiologist because we get a lot of advice from him. And I also want to thank Jeff, because Jeff is a friend brought me into the international society. He introduced me to many friends in abroad and invite me, and Christina invited me to the UIUP. We had a conference a few years ago. We met Jeff in many conferences, but I will to show my great thanks to him for his help and introduction. Actually, we had a lot of we have several papers is already published. Just recently, we have an MBL paper published about the blank soldier flag. So thanks again for the organizer to ask me to give the talk. And if you have any question, I will be very happy to give the answer. Thank you.
Thank you, Professor Huang. Questions? Welcome now, please.
Thank you very much, Yoni, for you help me to arrange the slides.
Of course. I want to ask something.
Yes, please.
Have you tried different methods of genetic engineering like HDR, site-specific integration, this type of method in your laboratory? Have you tried this? Have you have any experience with that? This is my first question. The second one, PTTH takes 300 days to pupate. How can you maintain such time? How is it possible for you? Do you have method to do it?
Yes. Yes. So you have two questions. You finish? Okay.
Yes.
Okay. So the first question. So we are doing the insertion of the system. So we are trying to, but still we are not success. So we are using the talents at the moment because we success using this in the silkworm, but still we need to try. So this is first question. The second question is about the PTTH mutants. So we keep it's more than 300 days now. When we want to keep it, we we inject ecdysone. We inject ecdysone, then we'll have the pupate. Then we could get the next generation.
Okay, thank you. Any more
Can
questions?
I ask a question?
Please.
Hi. So thank you for this very interesting talk. It looks like a lot of work and very, very successful achievements. I was wondering whether you looked at the interaction between the black soldier fly and the microbiome and the relation between the black soldier fly genetics and the association to certain microbes.
Very good question. Actually, we have a group working with microbiology research with blank soldier fly. You know, our blank soldier fly, we have the microbiology from two aspects. One is outside of the blank soldier fly. Another is inside in the guards of the blank soldier fly. So we have a group of students. So they are trying to understand the blank soldier fly and the microbe to make the derogation of the antibiotics. So we had a paper just published recently. If you need, I can send the paper to you. And also we so his test about this in the different development stage, the variation of the microbes within the body and outside and in the environment. So we know that this microbiome is very important. Without the microbiome, the black soldier fly cannot grow very well. So also, our student is trying to do the genetic modification of the microbiome. So so we have the so he developed a system using this microbiome engineered microbiome to knock down the gene of the black soldier fly. So he do success. So I think we are write this paper, so we could share these results with you. I think in the next conference or in the papers.
Okay, thank you very much.
Okay.
You're welcome.
I think because of time limitation, we will shift into the second talk. Thank you very much, Professor Huang, for your great talk and your great willing to participate.
Thank you. Thank you. It was really
Thank you very much. Thank you all. Thank you very much.
Jeff, are you ready for start?
I'm ready. Excuse me. Yes, I'm ready.
Okay, you can share
Okay.
your screen.
How's that?
Great.
Excellent. Well, first and foremost, Yongping, it's great to see you. It's been too long. Secondly,
Yes.
I just want to echo some points that Yongping made, and that is the success that we see with the species is truly about collaboration. And I am not a molecular biologist by any means. It's partnerships with individuals like Yongping or with Christine Picard and others that allow us to merge ecology and molecular biology together. And I've noticed in my career over the past 25 years of working with black soldier fly that while I'm still heavily engaged in research, I've really enjoyed this opportunity to network and bring new individuals into the field. So the purpose of this talk is really to talk about how the black soldier fly as a model can truly serve as a platform to explore basic research, but also answer applied questions. And that's important. I think we all recognize from a research perspective, especially when attempting to engage federal agencies for funding to support this work. And I want to say that also when I look at the individuals that are listed here, I was scrolling through to see who was present. While I truly enjoy the collaborations that I have with Yongping, Christine, and others, I love it. I look forward to working with other individuals like Jonathan, Yuri, and Yoni on future work. So if you're interested in learning more about the ecology side and how to bridge what I do with what you do, please don't hesitate to reach out to me. I'm always open to collaborations. Now, when we think about black soldier fly, what's interesting is this image is actually serving two purposes. Yes, it is only half the fly, but it is to indicate something, and that is we really know practically nothing about the wild fly. We know very little. Everything, I'd say 98% of what we know is what has been generated in a laboratory setting. So we really know little about how this fly actually operates in nature. And I think that's a huge opportunity for us. And we see the momentum that's building up for the species globally, which is amazing and it's wonderful. But, and while I've been part of a lot of that, I really want to, let's see if I can get this to advance. I really want to thank Dr. Shepard. I started working with him in the 90s on black soldier fly, but his work dated back to the early 80s, like 81, 82. So he was probably working on the species in the 70s. And this is him and his wife, Sheila. And we still remain good friends to this day. But back when people thought it was even more outlandish to think of this insect as a means to recycle waste and produce protein, Dr. Shepard never lost faith in this opportunity. In fact, I should point out that he approached me about working with the black soldier fly in the mid-90s. And I did not take that opportunity. It was a second opportunity that came to my table that let me work with it. So I'm grateful that Dr. Shepard gave me a second chance to work on the species. He's since retired. I do talk to him occasionally and keep him updated on what this industry is doing, but really he served as a really major driver of the industry from the very beginning. So I'm grateful for what he's done. So as you can see in the title, I talk about who, how, and why we're doing it. And I'm going to try to set that stage of what opportunities exist from a broader impact perspective of the industry, what that means to grant writing and pursuing funds. I had the opportunity to go to the Drosophila conference. And while I was there, I got to witness or observe the diversity of research that's taking place in that arena. And I talked to Aaron Tyrone, not this morning, it should say back in February, but I talked to Dr. Tyrone about this. He's a colleague of mine here at A&M, and he said, can you imagine all of this research taking place with black soldier fly? And I had two responses. The immediate was, I was overwhelmed. The second was, oh my gosh, that's huge. We could do really good things. So the title of my talk is The Black Soldier Fly, Hermetia illucens, Model for Circular Economics and Bridging Basic and Applied Research. And part of this will be discussed from the perspective of the Center for Environmental Sustainability through Insect Farming that is co-directed by Christine Picard and Heather Jordan. Here's my contact information if you have any questions about the presentation or our opportunities to collaborate. So we know there are model organisms that already exist. Drosophila is a key one that we talk about, but it is only one model organism. And if you were to talk to Christine Picard, she likes to use a quote that says, while it is a model organism, it's really unique in terms of its biology, and that exploring other non-traditional model organisms could allow us to explore broader questions, but also have more significant impact in different arenas of human health. So how can we use the black soldier fly to not only understand human immunology, health and nutrition, but also to explore with the black soldier fly clearly is how does that impact the environment, especially when we consider what we're dealing with today with climate change. So the black soldier fly, this one species out of about 2,800 species within this family of flies that's globally distributed, actually has some really unique sister species, but it can, I think it can apply, what we can learn from this one species can apply across different taxa. What's fascinating about the black soldier flies, we never think about it as an extremophile, but it is. It colonizes decomposing material. It lives in a highly toxic environment where it's exposed to numerous microbes that are competing for resources and potentially could harm it in terms of its health. And as Yongping pointed out, the antimicrobial peptide profile of the black soldier fly is massive, and is for good reason. But we also understand that this insect can consume a diverse repertoire of waste streams that range in nutritional value from next to nothing to really substantial. So how is this insect evolved to have the plasticity to consume such a variety of foods? And I think that bridges nicely with human health. I mentioned initially about the microbe-insect interaction aspect of it. Its microbiome allows this insect to not only inhabit toxic environments, but also take resources of low value and convert them into insect biomass. From the adult perspective, we can explore questions that relate to its ability to locate a mate, as well as how it takes care of its young. And you're probably thinking, well, it doesn't. But it does. It selects resources on which its offspring can survive. How does it do that? With the adult mating behavior, clearly that's excellent opportunities to explore sexual conflict or sexual selection. How do these individuals find each other, copulate, and produce fertile eggs? And this is an area that my lab has focused on for the past probably 8 years. And in the end, we know this is important from a sustainability perspective. We know this insect is very good at recycling waste. And one thing we never consider, or we rarely hear people discuss, is the black soldier fly is an excellent model to engage the community on the importance of sustainability and being proper stewards of the environment. So the black soldier fly is great for engaging the public, either in elementary schools or even in public forums where you engage adults. So why are we doing it? I think we all recognize as the human population is growing, we have to be better with the resources that we have, and they are really limited. And we know that as other industries are growing, such as the aquaculture industry, an industry that we spend a lot of time talking about, this is tons of production on the vertical axis over time on the horizontal. And we see there's an increased reliance on farm fish. So we know that if we can produce feed for farm fish using resources that are of limited value, we can become more sustainable. What's fascinating is that while we want to produce more food and we can produce more food, we waste quite a bit of food. So according to FAO, 1/3 of all food produced around the world is wasted. In fact, when we think about waste production, 43% of it occurs in our homes, 40% in restaurants, and 16% on farms, 2% in manufacturing. But the keys here, obviously, we can talk about how to engage homes in recycling waste or restaurants. But if we circled farms, I think that's an opportunity that we don't discuss enough. And that is when we produce food on a farm, there are waste products, especially when we harvest from the fields. How do we actually pull that waste from the field and recycle it with the black soldier fly? When we think about the aquaculture industry, again, the thing we should recognize too is it really is not very sustainable at the moment. So what do we want to do in terms of producing the feed so that we can farm fish? Well, the idea is, again, that we can use the black soldier fly to recycle these wastes to produce the protein that can then be used as a feed for aquaculture. But we can also extend that to poultry, swine, pets, and potentially even humans, if we think about the protein as an ingredient or the fat as an ingredient in the food that we consume. And there's growing federal, state regulations on how to manage our environments. And for example, in California, USA, many years ago, they passed a law saying no food waste in landfills. So there's federal or state pressure on how to be better stewards of the land. And that creates opportunities for the black soldier fly, because what we know is that the black soldier fly can reduce all of these different waste streams. Anytime you see a citation that's in red, that's a publication that I had the benefit or the pleasure of being a part of. And it just shows you how we can diversify our engagements globally. So we know black soldier fly can recycle food waste. We know that the black soldier fly can engage microbes and can suppress pathogens such as E. coli and even salmonella. And that's just the beginning. I mean, these papers are now 15 years or older now, and this was some of the initial work showing these interactions. Now we know that they can break down antibiotics. We know that they can kill antibiotic-resistant bacteria. And this process of recycling waste actually filters out antibiotic-resistant genes in the population. This paper has since been published when developing this talk. One thing that we've been exploring in my lab, and this is in collaborations with Heather Jordan at Mississippi State, also a co-director of the NSF Center, is that we are exploring the microbe-insect interactions, and we find that starvation impacts the microbiome and gene expression. Now, a question that we have is, can we manipulate the microbiome to induce additional feeding by the fly, the larva? In other words, make the larva feel like it's hungry and it will eat more. This can do a couple things. It increases conversion rate of waste, but it also increases production of the facility. And what we're finding is this is resulting in intellectual property. From this work, we're able to submit a patent last year. One thing that's fascinating about the black soldier fly, this work dates back to about 2014, is that we found that we can take the insect and actually put it into a hibernation mode at room temperature. And what you see here is on the vertical axis is time from inoculation to harvest. So once you place the larvae on a diet, how many days does it take to you harvest? And what you see on the horizontal axis is with this technology we developed, you can set the insect, the larva, at room temperature out to almost 65 days, and they will not grow. But as soon as you put them on a resource, you can harvest in 7 days, which is amazing. And that's with no manipulation, no molecular or microbial manipulation. It is straight ecology. So we can actually do long-term storage with this insect at room temperature, which is pretty phenomenal. We're also looking at, well, what is happening globally, because a lot of the work we're doing is with the Texas population, which in fact we all recognize as the shepherd strain. It's the population that we established in 1998 from Alma, Georgia. So this population has been used around the world, but we have to understand, because it's a global species, is how the biology changes depending on the population that we're working with. And in the end, what we want to think about is, how do we maximize waste conversion to produce protein that can then be used as feed for a variety of different livestock and humans? But it will be population specific. So we have to recognize that because of the genetic diversity that exists in the world, is that the results generated are specific. We don't know if these results translate globally. In the end, you know, we recognize that if we can integrate the black soldier fly as part of the agricultural cycle, we can create a more sustainable process of producing traditional foods and feeds, but also recycling that waste to enhance the product values being generated, which could bolster agriculture, while at the same time protecting the environment. One thing I love to point out about the black soldier fly, it is not owned by one individual, one country. It is a global species. This species can be engaged and applied anywhere in the world. And I love that, because that means that in global South nations, this insect can be used to bolster the economy, protect the environment, and decrease reliance on importation. And we know this has taken effect because we see the success that companies are having in the world, and that there is investments taking place in terms of growing this industry. But the thing is, we're still at the beginning. Companies such as Darling, Cargill. They're partnering with individuals, companies like Protix or Novafeed, et cetera. But they're coming together. And this is important from the application side. But there is that bridge that is really important with research. A lot of these agencies from the United States are coming, they're coming full circle and recognizing the value. And a lot of it is because of the applied benefits. But at the same time, I want to emphasize, there are basic questions that can be addressed with the species. These are agencies that I've been successful in getting funding from, but there are others that are still making that connection, such as the National Institute of Health. Hopefully in the near future, we'll see more efforts in terms of engagement in this arena. But that led to the creation of the National Science Foundation Center for Environmental Sustainability through Insect Farming. Again, to point out that it's co-directed by Christine Picard and Heather Jordan. We collectively have been working on this process since 2018 when Christine organized the first meeting at IUPUI, her university in Indianapolis, where Yongping attended and many others. That led to this concept, which was funded 2 years ago. And you can see the diversity of companies that are engaged. And this is an old slide. More companies are becoming part of the center. The beauty of the center is its low indirect costs, where 90% of the funds generated by the industry go to research, and you have access to all data generated. These companies not only have access to the data generated today, but the data generated yesterday. So the return on investment is about 47-1 in terms of value. So in the end, what we want to do is build that bridge between basic and applied research. I hope that those that are in attendance that are considering the black soldier fly as a model can see the benefit from a broader impact perspective of what this insect brings to the table, and we'll partner with other entities that are present or not in terms of engaging in research from an ecological, molecular, or microbial perspective. But we can do this to recycle these wastes and produce feed. So very specific applied values. So if we go back to that comment, can you imagine all of the research being done on Drosophila being done with black soldier fly? Yes, I can imagine that, and it's happening. We're seeing it. Yongping presented a wonderful presentation already about basic research opportunities that can be done. With that, I want to thank the funding that was provided through NSF and these other agencies, as well as Texas A&M, that have allowed me to do this work. I want to thank my collaborators. Those partnerships are critical for this industry. They're critical for understanding the species, and that will allow for accelerated advancement in a very responsible manner. And in the end, I have to put the statement up. I don't represent NSF, anything that I said. And I want to thank my lab, because the research is done by them, not so much me. And without them, I would not be able to do it. So thank you very much. So let me stop sharing. I can figure it out. But if there are questions, I'll be glad to answer them while I'm trying to figure this out.
Jeff, I have one question.
Yes.
Can you see based on your experience, the next insect model species that is not the black soldier fly? Like, can you see other opportunities that can be taken into the industry?
Yes, definitely. So that's a very interesting question. Are there other insect models that potentially could be integrated into the insect as ag industry? Yes, but it's a chicken or the egg kind of question, because it's regulatory driven. So there's been a lot of advancements with black soldier fly and approval, which opens the door for the efforts. We need more Dr. Shepherds that are willing to lead the charge on developing new insect models. And I'll give you a couple examples. Housefly. We would consider it a pest species because of its ability to serve as a vector for numerous pathogens. They're synanthropic. They have a lot of issues. But we got to think about it from a global perspective. One is that if we industrialize it and manage it, it's not a pest. Keep in mind the black soldier fly historically was a pest species. We manage it to prevent that from happening. The same can happen with houseflies. Secondly, blowflies that are very good at recycling meat-based products could be industrialized. There are papers on it already out there. And that's from the feed perspective. If we think about the food, obviously mealworm, cricket are out there. I'll give you an example of an industrialization insect that could be amazing from Jeff Tarnow's perspective. There is a species of soldier fly that lives in hot springs of Yellowstone National Park, USA. They live in water of a pH of 1, temperatures that reach 60 degrees C, arsenic 30 times the level that will kill an invertebrate, no oxygen. Imagine developing that insect to recycle.
So it's insect or area? It sounds like it's a area.
Imagine using it for bioremediation of toxic environments. We've not even begun to explore those areas. So yes, I do think there are numerous insect species we could work with.
Thank you, Jeff.
So Jeff, this is Yulia with Interproject. I have a question. First of all, thank you for the great talk. So what does it take to make BSF industry economically viable? What's the major bottleneck right now? Because we do see several large companies, but it's not widespread, it's not as far with other protein sources. So what do we need to do?
So I think it's a combination of factors. I think we see companies like Freezum, Protix, and et cetera that are working on this. Is how do we minimize expenses through the colony side, through the bioconversion aspects. Obviously, engineering comes into play of how to make it more efficient, increase the investment of production, but also engaging regulatory bodies or federal governments on how to create carbon credits or to encourage the use of this system. So I do think it's a combination of factors. The biggest is driving down costs to create a product of that can compete with the fish meal, soy, and maize commodity groups. And I'm going to pause on that statement. I don't want to say compete. How we can work with them to produce the feeds that are used. Because if we think about, I think that's also a major challenge, how we convey this model. Because I don't want to say it scares other industries, but it definitely gets their attentions. We don't want to say we're going to replace soy. We don't want to say we're going to replace maize. Those are big, powerful entities in the world. We want to work with them. So we should be advocating, how do we work with you to recycle your waste to produce products of value? So if we can partner with these other commodity groups, their influence could actually be quite beneficial for developing the industry. So a lot of different factors at play here. Driving down costs is a key one. Diversifying the industry. Does a company have to do it all? Produce the insect, collect the eggs, digest the waste, produce the protein? I don't think so. I think companies can diversify. I think there's a spectrum of what companies can be. There's no one right answer. I think there's a lot of different approaches to the model. And that's exciting because it shows you where we are. It's at the very beginning. And we have to determine what works and what doesn't, why it doesn't work, and how we make it work. And I think all of those factors play a role.
Thank you.
Yeah, my pleasure.
Any more questions?
Hey, Jeff, I have a question. So I was wondering, what is your opinion about co-culturing of the black soldier fly with other insects in order to promote like more efficient reduction of or better FCR and reduction of waste, which is not very efficient using the black soldier fly only?
I think that's a wonderful question. I think that also question demonstrates the niche that exists. If we think about companies like Hendrix, who can actually do the work to bring the different strains together and find out what works together to produce these novel strains. And then we industrialize and we have these strains that are used in the world and we know them. So we know why they work, why they don't work. And we can actually, these entities can work to solve questions when there's colony collapse or other issues. I think it's a great idea. I think I would love to see that company exist.
יגאל, שומעים אותך. Okay. Jeff, thank you so much for this talk and your participation in this webinar.
Thank you, Jeff.
Thank you very much, Jeff.
I just want to say thank you for the invitation. I think you all know I'm passionate about the black soldier fly. If anyone on this call has a question, if I can help, I will. So do not hesitate to reach out to me. I look forward to hearing from you. And Yuri and Yoni, thank you so much for this opportunity. It's been wonderful.
Thank you. Thank you very much. Okay, Eric, the stage is yours. Thank you.
All right, I guess you can hear me. Cool. So yeah, I think definitely one of the reasons that I was happy to get the invitation to speak and agreed to it was because I think, like Jeff said, the need for collaboration in this area is kind of really important and really exciting, and I just wanted to, you know, participate in that. So yeah, I'm actually pretty excited to be here. I feel like this is one of the most important areas that the industry will be working in the next, 10 years. So we wanted to share some of our thoughts and experiences. And, if the first talk really felt like you had a scientist and the second one felt like you had a visionary, then maybe this one's going to feel more like you're stuck with a farmer. But yeah, Kriti, who is on the call, and the head of our genetics group, and I did want to share some of the experience about actually industrializing an improved strain and developing it. So I'll walk you through. I'm going to try to be quick enough that we can have a relatively long time for discussion, because I'm going to try to lob some questions back at the group, which might be a nice way to wrap up here. I'll introduce Protix, then I'll talk a little bit about the history of the genetic program that we had, and then spend some more time on goals and considerations before kind of teeing up for a discussion at the end. Okay, so I'll be pretty quick here because we have 2 minutes, self-imposed, but that's our factory. We've been producing for a number of years. This is since 2019, but we had a pilot plant before that. Solid enough revenues in 2022. It's hopefully going to be a lot more this year if things don't explode in the next few months. Relatively decent amount of investments in R&D, pretty multinational, and we're really trying to sell in a lot of different locations and understand the markets there. We say that we're the leading insect ingredients company. I'm not going to push too hard on that, but I think that a major thing is that we really have been working to build the market to use these products, and since 2019, I think that we have probably supplied a good chunk of that market with refined protein meal, and that's given us a lot of insight into what customers are interested in and how to produce. And now, after doing that for so long, we're still interested in kind of, I guess, unleashing for the potential is the slide. So really within this sort of full value chain that we work on. So I think Jeff is right. I think that there will be time where segmentation occurs. But right now, at least one of the, I think, nice things about being a Protix is that we do everything. We have the full production process, including, genetics, entomology departments, diet formulation. We're really using a lot of that as we operate, a lot of that R&D and tuning on it. And then, a lot of work with customers on trying to find out what's the value in products. I think, the person who asked, what will it take to make the industry profitable? I think we're actually pretty optimistic about that point pretty soon. But a lot of it comes down to grinding out the economics and running a very tight ship and stuff. If you look at this picture, I mean, people are in front of the machines, but I'm, really just very proud of the machines at Protix. We really try to take very good care of those machines. So you can't really see them that well, but they're in the background. And I think, it's just been super fun working on them. Yeah, and then again, we work in 4 areas for the most part. So these sort of protein, lipids, kind of more raw products, and then plants. And plant, I'm actually very excited about. I won't go into too much detail, but these interactions between insects and plants are also an area that I think some quantitative genetics research would be interesting. When plants interact with the insect metabolites in the frass, we see some pretty astounding stuff, and it would be cool to see what's being upregulated and things like that. Yeah, some flavor. Okay, hopefully that was fast enough. Well, we started a genetics program back in 2017, and we did it with Hendrix Genetics. I think both companies were really interested in seeing what the species could do. And I think it was a really nice pairing. We wanted to, kind of make the steps forward with selection programs. So we did selective breeding instead of modification. But it was just really great to work with a group of researchers who have done this before with other species. I mean, Hendrix has made mistakes in the past. They've learned from it. So that as we were starting to come up with ideas, that we didn't, start to walk down one route and end up dead-ended because of something we didn't foresee, where, an experienced party like them would say, yeah, this kind of happened with chickens before, or, this is a known risk. At the same time, I think Protix has been really good at a few things. One of them is just study of behavior manipulation through technology. And it was just a lot of fun to bring that together. We built a pilot facility for the genetics program that's different than our other facilities. And then as we were doing genetic selection, we worked a lot to kind of tune what we were doing within our own production facility. And again, I think that this sort of full value chain situation we have at Protix right now, I'm really happy about that, because it lets us, identify where things might go wrong with a strain that does well in the lab but doesn't do as well in production. And I think after about 3 years, we felt kind of confident about the results that we'd achieved and the risk level of introducing this thing. So we published a paper. And this is kind of a hobby horse for me, but I think that, a lot of the work that many of you are doing now, I think it is transformational for the industry. And I think it's really important to share that with a general audience in a validated way, instead of just making claims. And we really wanted to put something out there saying that we've achieved long-term artificial selection in an industrial setting, so that we could rely on this to say that the genetic improvement is real, but also in a factory, but also other parties, including your companies, could say, hopefully when you're going for funding or something, what we're going to do has already shown results. We can do it better. I'm happy if you take the floor on that, but that, it's already becoming a baseline that the work that researchers like us are doing can make a difference in an economic context. So yeah, we published that, and I think the information here is pretty much the same there. It's old. We've continued, but I think that it shows really the promise of what everybody here is doing, because it's been now rolled out in our factories. We've definitely benefited from this. We've conducted that production with improved animals. It definitely shows better performance. We get a better capital efficiency. And at the time when it came out, we were looking at 40% bigger larvae. I think we all know now that you can go much bigger, but yeah, and I think that another really exciting thing for me was not only achieving this sorts of improvements, but also eventually disseminating it into a production facility and trying to work with operations staff to find out, what was the right way to grow this. I remember the first time the genetics team introduced these animals into the factory, it just didn't work. We really spent a lot of time working through all the risks, working through all of the different operational methodologies, training people, getting feedback, and collecting information so that we could do this again more efficiently, and also that we could improve the way that we do selection. So, we're through that exercise of rolling out a strain. I think now we feel more comfortable doing that more generally. And continuing to do improvements. And obviously we have some other goals going forward. And I think that these are, I'm sharing these mostly to give maybe some weight towards some of the research topics you might be weighing. But we're really focusing, of course, on environmental and economic goals. So, at the core of any genetics program, you need to deliver healthy and superior animals. So I just spoke about this dissemination activity, but a lot of focus for us as a producer is actually how to take the improved animals and make them work for an operational facility. Not only are they better, but that they can be used. People around them know how to use them. That the software operating system for the factory knows how to help people manage it. And on top of that, we're interested in trying to do things like, see how genetics could reduce energy usage. Improving the FCR, which I think is something that everybody has in their mind. I hope so. And then better health for the insects. And I think somebody mentioned disease at some point as an area where we might want to do more research. As we run our strain in our factory, we're built up to collect a lot of information on it. We can see, different ways that they hatch, different ways that they consume energy. And a lot of that is being monitored, and then we're looking for ways to validate that through the noise of production and stuff. So for me, I think that as I said, I think one of the biggest topics is how do we get to an improved feed conversion ratio on a general feed set would be great. I know that there's a lot of interest in tuning strains to specific feeds, and I think makes a lot of sense. But it would be really great to dive into what might lead to generally improved FCR. From an environmental impact standpoint, this would be great. I think even if you're using waste which emits greenhouse gases, it's still nice to be more efficient with that. You can then build a bigger factory and use it to produce more protein for people. But there's really an interesting environmental aspect to a lot of the genetic work that everybody here is doing, which I hope we all reflect on, ways we can direct our work towards that. So I just did some stupid math on an envelope, but if you look at broilers, which is kind of an analog of us, it took them about 30 years of selection to cut the FCR in half. If you look at the black soldier fly lifespan, I think it would take less than 7 years because of the time it takes for the two species to reproduce. And then, I'm obviously very curious with the work that we're all now more and more interested in with modification. Could we get there faster? Yeah, so I think as a company, this has been something that we've weighed over the years. What is the role that we're taking by selecting a strain? And then, for some reason, I think CRISPR sounds like it's another step forward, even though I realize that there's a lot of arguments you can make that CRISPR and selective breeding are not really yielding such a hugely different result in many cases, if you just look at the genes at the end. Potential for this is huge. I think we all see this. At the same time, we're already being scrutinized as an industry for ethics and the welfare of the animals, which is why it's also interesting. I think some companies that work in genetics really feel like, if they're going to approach genetic modification, they're only really going to do it for ethical issues right now, like illness. So for some companies, they'll make an ethical decision. We are willing to do work like this if it makes the animal's welfare improve. Otherwise, we don't feel comfortable with that. Another topic that I'm very interested in is how can we use techniques like this to reduce the risk of inbreeding? Super interesting, major consideration for regular selective breeding. And then, again, I reflect on the work that we do, which is heavily leaning towards watching the behavior, using these strains in production. And while I'm super curious, I'm very confident that we're going to see selection and modification nail down key traits like FCR. We've already seen larger larvae, stuff like that in selection, in modification. I'm curious to see how we're going to modify the animals in order to take advantage of more complex metabolic processes or other complex processes, and where the interplay between selection and editing will come together. I think there's a lot of value in both approaches. We're definitely interested in working on both of these to kind of tease out complex interplay and then maybe accelerate with CRISPR or something if we can see that in the lab. Yeah, so those are some of the major goals we kind of have for the discussion. I think I do want to give time to talk about this stuff. I'm really curious in this group to just hear, what does your group do when they approach the ethics issues? I mean, I'm very curious how that is taken by a science, focused company where you're really in the lab almost all the time versus a company like Auschwitz has labs. We also have factories, and we're also working a lot with other farmers. How do you approach the ethics issue? How do you communicate? What kind of challenges do you face going from the lab to the factory? And where do you think the biggest win from quantitative genetics will be? I mean, I think I named FCR, but I'm interested if people have other ideas. So I think it's just an opportunity to reflect on kind of the industrial context I'm coming from, but then I'd like to hear your own insights on that. That's it. So the floor is kind of open for discussion, I think. Any questions first to maybe get things going?
Eric, this is okay.
Right. Sorry.
I go first.
Thank you, Eric, for your very nice presentation. And with all your experience in breeding, I was actually wondering if you have ever noticed some phenotypical difference in the breeds, such as like new patterns or colors or eye colors.
You see phenotypical differences between the animals.
Can you just repeat the question?
She's asking if we've seen phenotypical differences between the strains. You definitely do. I mean, I think obviously one of the big phenotypical differences that people see is things like size. We've definitely noticed different phenotypes like colors while working with the animals over the years. We were very curious for a while. Could we exploit that? And maybe that's something that could be exploited by a company that would be willing to do modification, because you could use it as a marker or something like this. But it was not a priority for us. But there are different phenotypes on colors, for example. And then I think the main phenotypes that a lot of the producers in this group would have tried to see are classic production characteristics.
Thank you.
So I have a question about the selective breeding. The way to go is like people are doing all the poultry working, like having grandparents and keeping different stocks and combining them, or from the beginning you're screening for one all the time for some trait and then keep on doing it and you have only one strain. Is it better, not better? What is the best?
I think it depends on your goal. I mean, we've had different strains, some continue, some stop. I think some of them are there to, at this point, de-risk engineering choices. I mean, I don't really care to have a strain that's specifically good at something, but we want to even use the kind of processes we've built up while we've done the genetics program to improve our engineering programs. So it really depends on your purpose. Some of them will never see the light of the day, or they'll be stopped. Some of them will continue. Some of them will be interbred. I mean, genetics is really just it's the program running a lot of the animals. So you see them in different contexts. Sorry, go ahead, Anna.
Do you have any like inbreeding monitoring programs?
Yeah, we do. Yep.
So you monitor your population like over time.
Yep. But I would love it if somehow we could modify animals if inbreeding happened and avoid this forever.
Yeah.
If inbreeding
it's
just like turn back the clock or
difficult.
something. Like for a while I was and maybe in the more discussion framework, I was very curious about the freeze zone technology to freeze strains. But I've started to wonder, like, if you can just edit the strains, do we even need to freeze them anymore? Or can we just kind of roll back the software, so to speak, if we want to? So, I'm super interested in that as a way to back things up. If you just, know the sequence, can you go back and forth in time purely with the data?
We can surely discuss it.
I would love to. I mean, I'm here partly because I realized that we're going to all be working on stuff like this, and it's all going to be out there. So it's interesting to discuss these sorts of things, and then, I'm interested to know what's possible there now or that we think is possible in the future. But yeah, for now, inbreeding is a risk, and you gotta watch for inbreeding. Sorry, Julia.
Yeah, finally. Thank you for the great talk. So I have a question about the output of the product. So in this improved strain, so you mentioned that actually you've witnessed increase in FCR and other parameters. Have you seen any changes and alterations in the product output, fat to protein ratio?
I didn't say anything about FCR, even though I kind of want to. But again, you know, the slide I said about publishing the results and stuff, I'm pretty conservative about claiming anything. I really want to see anything go through our factory a few times. So for now, if other parties have seen it and they can claim that super reliably, that I think is great, but I want more data to prove it. Just as a caveat, but I think FCR is a major area to work on. Change in composition, well, we didn't really have it. We monitored that really carefully during the program. So that was something that we wanted to de-risk. There's a lot of risk in these strains. This is something that Hendrix beat into us. So we were watching this from the beginning.
Okay, thank you.
Eric, I have a question. I've got to run for another meeting, but how fixed are the traits?
You mean if you stop selecting, do they disappear?
Yeah.
Well, some of them seem pretty steady, I have to say. Like, I think that the thing is that there's a lot going on in an environment, and what you'll see is that a trait might kind of stay, but it might be eclipsed by other things that start to dominate it if you don't kind of keep things in line.
That's great. I think it just shows that it's complex. So I appreciate that response.
And if you have to go to the bathroom, you may now.
I got another meeting. Hey, y'all take care. Have a wonderful day.
All right, see you
Thank
guys.
you, Jeff.
Well, see you, Jeff. But yeah, more questions or discussion are welcome. I am really curious about how some of you see the ethical aspects or the important traits to work on and stuff. But more questions are also fine. We can keep going. Anybody else? Any other questions?
I have a question. If you can say anything about trade-offs, like obviously when you select for some trait, you can have a negative effect on others. Is it something that you see in specific traits, or like how do you tackle this?
Yeah, so I think there's sort of two kinds. One of them is, yeah, there's really a trade-off between two traits, and then there's the trade-off in a trait because of how you got another trait. So, you know, for that second one, that means really carefully designing the context that you work with your animals in and evaluate everything. Yeah, I mean, there are trade-offs between traits. I think that that's one of the good things about working with the Hendrix team is that they had seen a lot of those beforehand in other species, so we were kind of on the lookout for them. But you know, yeah, you want to start monitoring that stuff almost from the start. And I mean, we see that in the first talk. You know, they were looking at, you know, even as simple as if we remove the wings, does it affect fecundity? I mean, of course, but yes, there are definitely possibilities for that kind of problem.
So another thing is about the strength. So the strength that you are trying to generate, I mean, you generate, are they good for any kind of feed? I mean, not necessarily, I mean, like the fish will need something completely different than
so far so good. Yeah, that was a major, you know, for me, like one of the best moments of my research life was when we got pretty good genetic improvement transfer to different feeds. I was thrilled because, you know, you do work in a lab and stuff, and then you wonder if it will translate. I mean, there have been other areas where I think, you know, we have to do more, but yeah, it's been a pretty good transfer. I mean, it's at least worthwhile to use the improved strain on pretty much everything.
No, but I'm saying that you're using the same strain for any kind of feed for any kind of animal.
Right now, I mean, right now that's easier to do. Yeah, I mean, there's definitely going to be value in specific strains for specific feeds, but there's also a lot of management overhead to dealing with that.
Mm-hmm.
And I think that, you know, one of the takeaways I had is running multiple strains in a facility that's a production environment with human beings managing that is not nice. So, I sometimes wonder these companies that think we will buy eggs from 2 suppliers with different strains. I mean, that's pretty risky. Good luck running that factory. Seriously,
Yeah.
it can be really wild how different things are. And then just having them around. I mean, you could consider buying them in, but usually there's a pretty big multiplication step you'll need to do to feed a factory. So at least on your colony side, if you even have that capacity to jump between strains, is expensive and complex. So yeah, we wanted to get something that would be flexible enough to give benefits across scenarios that even ones we can't foresee. We didn't really want to develop something and then find out that the supply contract for that feed collapsed, which happens. I mean, if you're buying feed over the last 10 years or through Corona especially, you've seen factories open up and shut down. And, if we built a strain for one of those feeds, we might have been in a total loss on that. So yeah. But that's a design choice. I mean, and a choice of what you want to manage in terms of your spin up of strains and stuff like this.
So what about other products? I mean, like not only for protein. I mean, you talked about the frass and other stuff. What about the oil?
You mean in terms of breeding for oil, or?
Yeah, I mean, like doing something else. I mean, improving for that or other
I
things.
like the protein and its derivatives more than the oil. I think I'm not sure I would go all in on oil. What I was saying with the frass is more that I would be interested more in the understanding of what the frass is upregulating in the plants. I think it'd be really cool to see that interaction between, but that's a side show for this conversation. I mean, we're not talking about improving black soldier fly. We're just trying to understand, interactions, which seem to be pretty strong. So yeah, it's more of an interest there. I don't think I would modify the insects for the frass per se. At least not now. Yeah. But it's very interesting to think about stuff like, how can you use less energy or, other environmental impact vectors as well. Yeah.
We always deal with the ethics, all of us, it's not like we have a magic answer for everything. So we're all dealing with it some way or another, so each one on each is business and everything. So
yeah,
that's a huge thing that we have, I mean, the market or the government, we have somehow to teach them slowly and go through in order to.
I think that transparency is going to be really critical to that discussion. I think that, trying to publish our results, like I think, a lot of people in this audience are probably in the lab potentially modifying animals directly, but even selective breeding is an ethical consideration, right? I mean, you're changing animals anyway, so
Yeah.
we really wanted that to be public.
But
does anybody else have thoughts
but
on
that
the
will be, I mean,
maybe,
the market
maybe you and I need to have a coffee together soon. But just to give other people space to talk, if that's okay with you.
Yeah.
Does anybody else want to weigh in on the ethics topic? I think Yuri and I would be curious to hear your thoughts on that, or some of the other points that we brought up. We're all struggling with the ethics topic.
Idan from Frism. So first of all, one very interesting question. There were a lot of questions, but one very interesting question for me is like the challenges of taking the genetic improved strains from the lab to industrial setup. And so you said a few words about that, but if you can share a little bit more about what challenges did you anticipate to have, and what were the actual ones that you experienced, the ones that you are willing to share? And just a comment regarding ethics. So I think it's a main issue for all of us trying to modify in either way the genetics of the black soldier fly or like any other species. I think that as long with that our route will be transparent and clear, showing that all we are doing are within the framework of trying to having better solutions for for the environment while maintaining the certain set of rules of how to maintain the welfare of the animals and how to prevent non-essential harming for the well-being of those animals. It should be okay first for us, and then to communicate to the regulations and all other people that might be involved in the process.
Yeah, and I'm happy
Yeah,
to hear you
it's-
say that. I think the more that we reflect on that as a group, I mean, even if it's as simple as we just have to try to be transparent and stuff, the more we say that amongst ourselves, hopefully the more that we do it. And then, yeah, at least there's some place for people who have questions to go to as we build up the material. Yeah, challenges. Well, I think there's a lot of different kinds. I mean, I think, you know, we had 3 years to kind of anticipate them. So I don't know if there was anything that was really like shocking. You know, again, it was pretty interesting to see how the operations staff didn't know at all how to use these animals. We'd done a lot of it in secret, and so we kind of knew that day was coming, but it's still interesting to see people who have worked with black soldier fly for years kind of just be completely caught off guard by how a growing process can go. I don't know if anything was surprising. There's just a lot of work to tune everything out when you implement. And then the other thing is, yeah, throughout the whole process, trying to anticipate where you could go down the wrong path and screw things up very badly in a few years. And also genetic protection, all kinds of stuff. I mean, I don't think any of it's surprising. I think a lot of it's just like discipline and planning. Is there anything that was really surprising? It was exciting. I don't know if it was surprising, but it was exciting anyway. Sometimes it's exciting to see things. You probably experienced this too. You kind of know you're trying to do something, but it's exciting when it happens, even if you knew it might come. Yeah. It's still exciting to see these animals in the factory. I mean, it's super cool to see a giant building full of animals that have somehow been made somewhat that way. It's very interesting. Anyway.
Just another comment regarding what you said about the freezing technology and the way to modify each time the strains according to the needs after you know what you need to modify. So yeah, it's very interesting. I mean, there is no one answer, but one thing that you can do, like when you take generation after generation and you just freeze the strains in each generation, and you have like you can go in any step back to the generation that you think.
But I have a question. Since you're the guy for this, can I just ask the question? Would I need to, if I knew, you know, the distribution of alleles for that strain or something at that time, could I just kind of reprogram backwards if I wanted, or would I actually have to extract and redo that? I mean, it feels like if you just at some point, I mean, we're not there today probably, but if there was a platform for CRISPR and we were allowed to do it and stuff, I kind of imagine you could just say, let's go back to shepherd times or something. I mean, you could just put the stuff back in.
Yeah.
I mean, do you see that kind of time machine coming someday?
I think it's a very good question, but you know, you can basically, we are also doing CRISPR, and it's like things that you can do, but the ease of use and the amount of time that you will need to spend and the effort in order to have the exact strain with the traits that you want and the modification that you want, comparing or just taking outside from the fridge the strain and just continue working on that. So it should be easier with the freezing solution, but again, yeah, it is possible. I mean, it's all about how forward the technology will go to.
Yeah.
And
no, again, I say for now, I think that, you know, the frozen genetic material is really good, but I'm confronted by a lot of the work that we do and the interaction. I think that a lot of us are working on different things that are developing very interesting results on their own right now, and it's really worthwhile to pursue that. But as time goes on, the things we're doing will converge, and the only way forward will be to bring them together. Yeah. I see that we've done selection. We've been working on more quantitative genetics over the last couple of years. And yeah, I mean, the selective work feels incomplete now that we've worked on other things. I mean, you know, it all starts to inform itself. So yeah.
So how one question is, how can you maintain like the selection pressure while you are in that industrial setup? So as a follow-up to Jeff's question about the traits, and do you lose them throughout time? So it's, yeah, I think
I think we're very similar to we have a very similar approach to, like, you know, another genetics company where we have stuff in factories and then we disseminate. I think that's why we're so interested in not only getting to the improvement, but also the whole chain of getting healthy and superior animals into facilities and stuff, and training people and all of that kind of thing, because that's just, all of us are going to have to do that. I think there's, yeah, no good way to get around updating the whole context around a new strain or an improved version of one. So yeah.
Okay,
yeah,
guys,
and I don't think you necessarily want to do it in a factory.
Guys, I suggest we will continue offline because we are running out of time.
Yeah.
Okay. Thank you very much, Eric, for the talk. Everybody that participates, the great speakers. As I said, you are welcome to follow our activities in the website and in the LinkedIn page. And have a great year, everybody. This is the new Jewish year, so we wish you all a great year, and we'll be in touch. Thank you very much.
Thank you for organizing, Yoni.
Thank you. Thank you.
Thank you,
Yoni.
Take care.
Thank you. Bye, guys.
Bye.
Thank you. Bye-bye.
Thank you.